Hi students, today I am going to discuss about the Mendel's principle of inheritance. In my previous video, I have discussed what is monohybrids. Okay, how this monohybrid crossing is done. After doing monohybrid cross, Mendel was able to observe certain factors. Okay, so certain statements. These statements he has collected and given as a principle of inheritance. Okay, the monohybrid or pair of contrasting trait எடுத்து crossing பண்ணாரு. So crossing பண்ணப்ப hybrid கிடைக்குது. இந்த hybrid so அவர் எடுக்கும்போது அந்த parent ஒவ்வொன்றும் ஒரு pure breed. ஸோ இந்த ரெண்டு பியூர் பிரீடையும் கிராசிங் பண்ணும்போது F1 ஜென்ரேஷனில் அந்த ஹைப்ரிடில் ஏதோ ஒரு கேரக்டர் ஒன் ஆஃப் திஸ் டூ பேரண்ட்ஸ் கேரக்டர் ஒன்லி அப்பியர். ஸோ ரெண்டு கேரக்டரும் அப்பியர் ஆகாமல் ஏதோ ஒன் ஆஃப் த பேரண்ட் கேரக்டர் மட்டும் F1 ஜென்ரேஷனில் அப்பியர் ஆகுது. ஸோ யூ தாட் தட் சர்டன் ஃபேக்டர்ஸ் of the character dominant over another factor. Okay, one trait is dominant over the another trait. அதனால அப்படி observe பண்ணும்போது so one factor, one allele is dominant over the another allele. So thereby he has come to the observation. So certain factors, that is certain factor which is dominant over the another factor, or the allele which is dominant over the another allele, that is able to express its phenotype. Okay, so on the express பண்ண அந்த allele வந்து dominant allele, which the allele which is not able to express itself in F1 generation, because this certain dominant allele mask over the recessive allele. So what is recessive allele? The allele which is not able to express phenotypically in F1 generation is known as a recessive allele. Okay, so after taking he want to do certain research in that. So thereby he continued with the F1 progeny and the F1 hybrid irukla other self pollination. He allow for the self pollination allow பண்ணிட்டு பிறகு அந்த seed F2 generation கிடைக்கிற அந்த seed வச்சு அதை வந்து germinate பண்ணி அந்த plantல இருந்து இருக்கிற அந்த seed collect பண்ணும்போது that he is able to easily identify in F2 generation லையும் recessive trait appear ஆகுது. F1 ஜென்ரேஷனில் அப்பியர் ஆகாத அந்த ரிசர்சிவ் ட்ரேட்டு F2 ஜென்ரேஷனில் அப்பியர்ஸ். ஓகே, ஸோ when that F1 hybrid is allowed for the self pollination, automatically in the F2 generation the dominant trait as well as the recessive trait both are appearing. Though this recessive trait is not able to appear in F1 generation, but it is able to appear in F2 generations. So based on this observation, he has come to the conclusion. Okay, the traits do not mix up. Okay, the characters do not mix up, do not blend with each other. So adu மூலமா அவர் வந்து disproved the inheritance of blending theory. The inheritance of blending theory. இந்த ரெண்டு கேரக்டரும் mix ஆகாம okay they express independently. In F2 generation is able to see that either it is a tall plant or dark plants. He could not identify the intermediate plant. Okay, so intermediate height of the big plants. So thereby he concluded. So this is disproving the law of blending inheritance. Okay, so blending inheritance is disproved by this law of dominance. Okay, fine, let's move on. What is that first law he has proposed? That the law of dominance. So according to law of dominance, when a pure breed, when two different plants are pure breed with a pair of trait, the trait is a contrasting trait. Okay, here you said only one factor express itself in F1 generation. Take the two pure breed of the plants with a pair of contrasting traits. Okay, when it is crossed the hybrid, the F1 gen, in F1 generation only one factor or only one allele is able to express its phenotype in the F1 generation. Thereby this in hybrid only one allele is able to express its phenotype. So thereby it is known as a dominant allele, which is not able to express itself in the F1 generation. That allele is known as a recessive allele. Okay, so the expressed allele is the dominant allele or factors. The suppressed allele, suppressed means recessive allele. The recessive allele is known as a recessive allele or a recessive factor. Okay, however, this recessive factor, which is not able to express in F1 generation, but it is able to express in F2 generation. So thereby in F2 generations, when the F1 progeny is allowed self-pollination, so it produces the F2 generations. The F2 progeny, in F2 progeny, both dominant and recessive traits are expressed. Okay, in the 3:1 ratio. 3:1 ratio. Got it? So this is the first law, that principle of inheritance, the law of dominance. Followed by the law of dominance, Mendel proposed another law called law of segregation. Okay, so law of dominance தொடர்ந்து இன்னொரு law வந்து propose பண்ணாரு. That's a law of segregation, or it's a law of purity of gametes. Law of purity of gametes. So what does this law of segregation explain to us? அதனுடைய கான்செப்ட் என்ன எக்ஸ்பிளைன் பண்ணுது? So when a pair of contrasting traits are brought in hybrid, so on the hybrid la kondu varumbodhu, pair of contrasting traits hybrid la kondu varumbodhu, what happens? So this pair of contrasting traits, the factors of the trait are remains united together without mixing, without blending to each others, without blending to each others, without mixing to each other. The pair of contrasting trait in the hybrids remain together, okay, and separate out at the time of gamete formation. They separate out at the time of gamete formations, okay, and randomly reunite to form the trait. Randomly reunite to form the trait. Clear? Do you understand what I am saying? In case of law of segregation, என்ன சொல்றேன்னா, அந்த pair of contrasting trait வந்து அந்த factors hybridல வந்து அப்படியே இருக்கு. Okay, they are not mixed up, they are not blend. Already I told you what is the blending inheritance theory. Mix ஆயிருக்கும். But law of dominance வந்து அதை clear எக்ஸ்பிளைன் பண்ணிட்டு மெண்டலோடைய அப்சர்வேஷன்ல F1 ஹைபிரிட்ல he only get a tall plants. அதே F2 ப்ராஜனில டால் பிளான்ஸ் கிடைக்குது, டார்க் பிளான்ஸ் கிடைக்குது. இந்த ரெண்டு தான் கிடைக்கும். அந்த நடுவில் அந்த மீடியம் ஹைட்டு டார்ஃபும் டாலும் சேர்ந்து ஒரு மீடியம் ஹைட்டு பிளான்ட் வந்து கிடைக்கிறது. ஸோ ஐ அண்டர்ஸ்டுட் வெரி கிளியர்லி. இதில் வந்து the factors in the F1 hybrid, they remain together simply. Okay, at the time of the gamete formation, the factors they separate out and randomly reunite to form the trait. So that was his concept. So here he explained very clearly, and that no blending is taking place in the characters in the plants, in the monohybrid. So thereby he is able to disprove the law of blending inheritance theory. So look at this, the statements. Okay, so why do we call it? It's a law of purity of gametes. So that trait, the factors never mixed up, they express itself, they express its own allele. It is not going to mix up and express intermediate form. So if it is a tall, then it is a tall allele, the allele for the tallness, dwarf, the allele for the dwarfness. So that can be expressed. That's why it is when a pair of contrasting trait brought together in a hybrid, they do not mix up, they do not mix up or blend, but simply associate and remains together. Okay, and they separate at the time of gamete formations, randomly reunite to form the trait. This is known as law of segregation or the law of purity of gametes. So Mendel wanted to continue his experiments with the two pair of contrasting traits. First he has done in the monohybrid only one pair of contrasting traits. Now we wanted to do two pair of contrasting traits, whether this one pair of contrasting traits is working out or it is preventing another pair of contrasting traits. So we want to know whether it influence one pair of contrasting trait influence on the other pair of contrasting traits. So we wanted to verify with that. So he has done the experiments called the dihybrid cross. Okay. So in the previous one that was monohybrid. In the second our follow up and experiment on the dihybrid. Dihybrid cross. So in dihybrids he has taken two different parents, pure breed. Okay. So with the two difference, two pair of contrasting traits. Monohybrid has crossed the two pair of contrasting traits between the two different parent. Okay, so here he has taken the characters. So example, first pair he has taken the seed shape. Okay, so round versus wrinkled. So second pair color, seed color. Okay, seed color as in yellow was green. So first pair of contrast trait he has taken the seed shape, round was wrinkled. Okay, second pair it is a seed color, yellow was green. So he has taken the parent, that is in round and yellow seed, crossed with the wrinkled and green. Okay. So now, so what is going to, so what is that? The factors for the ground are the yellow, the same way R, and this is a small letter y. Okay. The factors from the homozygous. Separate out, enter for the fusion during the gamete formation from that Y. So we can write it. So here also so what is going to happen? They fuse. This is in gamete. Cross pollination is done. So all these things. F1 hybrid, F1 progeny will be round and yellow color seed. Round and yellow color seed. Got it? Round and yellow seed. So in dihybrid, Mendel has taken these typical experiments. So he wanted to compare with the two pair of contrast traits with the parents, two different parents. Okay, he has taken two pair of contrasting traits. So want to know whether one trait is disturbing the another trait. Okay, so he has taken the seed. So in the seed shape and colors. So seed shape, the round it's dominant, wrinkle is recessive, seed color. Yellow is dominant, green is a recessive. Okay, so here he has taken the gametes from the homozygous. They randomly reunites. So separate out and randomly reunite. If we have one progeny, express the round and yellow seed. So F1 progeny of dihybrid express the round and yellow seed. Okay, so he is comparing with the monohybrid. Same thing. If you have one progeny, only one of that traits will be expressed. The dominant traits will be expressing out. So thereby that we get it the round and yellow. So he has taken this round and yellow. What are the possible gametes will be formed from this F1 hybrids? So F1 hybrids we got it at gametes. It is an heterozygous gametes. F1 hybrid we got heterozygous gametes. That is an R Y Y. R Y Y. This is an heterozygous gamete, F1 progeny. So what are the possible? So gametes, the factors from the trait they separate out. The factors from the trait they separate out. So in F1 hybrid, as I already explained in the law of segregation, okay, contrasting traits, the factors of contrasting traits, they remain together, associated together, but they do not blend. Okay, so at the time of gamete formation, they separate out and they randomly reunite. So thereby, so they separate out and randomly reunite. So what are the possibles? Now they are going to randomly reunite. What are the possibles? Gametes. 4 gametes are formed. Okay. So we can have R Y. The same way R Y. Again R Y. Again these are the possible combinations. So 4 possible combinations. Okay. So 4 types of gametes are able to be formed. What are that? Number 1, RY. Number 2, RY. Number 3, number 4, small letter R, small letter Y. These are the 4 possible gametes formation in the F1 hybrids of dihybrids. Okay. So now we are going to allow the self-pollinations. We are going to allow the self-pollinations of F1 hybrids. We are allowing the F1 hybrid for self-pollinations. Okay. So when we allow the F1 hybrid, that is in F1 self-pollinations with F1. Clear? So what are the possible gametes we got it? So we got it that possible gametes. These are the possible gametes. We got it. Am I right? We are going to make the products with the help of the Punnett square. So this is an male and this one will be female and female gametes, male gametes. Now here it is very easy to find out the combination of F2 progeny. Okay, now what you are going to do it? Randomly reunites to form the traits. Am I right? So RR YY. Again RR, capital letter Y, small letter y. Again or capital letter or smaller recessive, then we are having y. Clear? So next thing, the same. Next we have to do it. The same you can complete. Capital letter R. Always the dominant allele should be written in the first. Okay, so keep it in your mind. Always the dominant allele written in the first, followed by the recessive allele. Clear? So we made that F2 progeny. Okay, let's check over it. So F1 is allowed for the self-pollinations, because in F1 hybrids we are able to get 4 gametes and these are randomly urinated and forms the traits. Clear? Now look at that first one. So we have to take an capital letter R and capital letter Y. So how many capital letters R or Y is there? How many are there? First one, R, Y. So how many are there? Just check out. So we will identify the 9. Clear? Then followed by that next one, R, Y. So we will get it as 3. Clear? So we will get it in R, Y. So these are the possible plants are available in the F2. Totally 16 plants are obtained out of 16 plants. So first thing you see that round and yellow. how many homozygous, pure homozygous, round and yellow are available. Am I right? Clear? So these things, round and yellow, got it? Round and yellow, pure one. I am talking about the pure one. Got it? Y. How many are there? So round and yellow, you will identify 9. So 1. So look at that. Round and yellow dominance. Pure one, this is it. Followed by that here. So you will get it round and yellow. Yellow. How many are there round and yellow? So 1 round. This is 2. And this one 3. And this is also round and yellow. This one 5 round and yellow. This is round but it is not yellow, green. So this is round and yellow. This one round but it is not. Okay, this is also round and yellow. And this is also round and yellow. This is a ringled yellow. This is also ringled yellow. You will find it. It's also 9. So round and yellow, 9 plants are there. Round and yellow, 9 plants are there. Round and yellow are 9 plants. Got it? Clear? So first thing is over, round and yellow. Next things, round and ringled. Round and ringled. So next thing is a round and ringled. Sorry. Round and green. Round and green. So how many round and green are there? So this is over, no? This everything over. This is a round and green. So number 1, round and green. This is also a round and green. This is number 2. Okay. This is not. And this one also not. So this is yes. So round and green. So round and green, how many plants are there? 3. Round and green, how many plants are there? 3. Now ringled and yellow. Ringled and yellow. Ringled and yellow. Ringled and yellow. So ringled and yellow, same thing you will find 3. Okay. Then ringled and green. Ringled and green is 1. Okay. So now we are able to get 4 types of plants. So number 1 is the plants which is round and yellow. Okay. Round seed and yellow colors. Round seed and yellow color seed. Okay. So how many plants are there? 9 plants are obtained in the F2 progeny. The second one that you will identify, ringled and green. So 1. Ringled and green 1. So here see, round and green, ringled and yellow. These two or the different, it is not belongs to the parents. Okay, it is not resemble like a parents, pure parents what we have taken, because it's a combined round and green. The round color seed, round shape seed, and green color, wrinkled shape and yellow color. These two are the new traits obtained in the F2 progeny of dihybrid. We have taken the parent trait round and yellow and wrinkled and green. These two parents only we have taken for the dihybrid. But apart from these two characters, Mendel is able to get two more different characters, two more new traits, that is round and green, wrinkled and yellow. Do you understand what I am saying? Apart from the parental characters, that Mendel was able to observe two more different traits in the F2 progeny. So thereby he come to the conclusion, yes, one trait is not affecting the another trait. One trait is not suppressing the another trait. So these two contrasting traits are expressed individually and independently. Okay, so thereby he brought that statement, the law of independent assortments. When two pair of contrasting traits are crossed, okay, so each traits they express individually and independently without affecting the another trait. Individually, independently, without affecting the another trait. So here we have taken that parent round and yellow seed, round shaped seed and yellow color seed. So this is a one pair of contrasting traits. Okay, the round versus wrinkle, yellow versus green. So two pair of contrasting traits are taken. Okay, however, he crossed in the F2 progeny, is able to get round and green, wrinkled and yellow, the two more new traits. So thereby he concluded, the Mendel concluded, so one trait is independent of the another trait and express independently. So that's why he brought that the law called the law of independent assortment. One trait is independent of another trait and express equally and express in the F2 progeny. So what is the, I think you are able to remember the Punnett square of dihybrid, so I am erasing that one. Clear? I think you are understanding the dihybrid. So dihybrid two different pure breed எடுத்துக்கிறார், but he has taken two pair of contrasting traits. So இருவேறு கலப்பினங்கள், இருவேறு பண்புகள் எடுத்துக்கிறார். ஓகே, ஸோ இந்த டூ பேர் ஆஃப் கான்ட்ராஸ்ட் trait எடுத்து கிராஸ் பண்ணும்போது F1ல வந்து என்ன நடக்குதுன்னா, only one pair of contrast trait is able to appear. Only one character வந்து F1 hybridல appear ஆகுது. அதாவது he has taken the seed, okay, round and yellow. So round and yellow parentையும், clear, wrinkled and green அந்த இதையும் ரெண்டுத்தையும் கிராஸ் பண்ணும்போது F1 hybridல அவருக்கு கிடைச்ச அந்த ரிசல்ட் என்னன்னா ரவுண்ட் அண்ட் எல்லோ தான், ஹைபிரிட். ஆனால் இந்த ஹைபிரிட் வந்து ஹெட்ரோசைகஸ், டிஃப்ரெண்ட் அல்லீல்ஸ். ஸோ ஒரு அல்லீல் வந்து இன்னொரு அல்லீலை சப்ரஸ் பண்ணி அதனுடைய த டாமினன்ட் அல்லீல் ஆல்வேஸ் எக்ஸ்பிரஸ் த phenotypes. ஸோ அதான் நடக்குது. ஸோ அதை எடுத்து அந்த F1 ஹைபிரிட்டில் 4 possible gametes are formed. 4 possible gametes because it's an heterozygous. 4 alleles இருக்குதா? So in the 4 alleles, 4 possible gametes are formed, and the gamete in this hybrids they separate out, randomly reunite and forms that F2 progeny. So the F2 progeny, the 4 possible gametes வச்சு அது F1 செல் பாலினேஷன் பண்ணும்போது ஆட்டோமேட்டிக்காக என்ன நடக்குதுன்னா 16 பிளான்ட்ஸ் கிடைக்குது நமக்கு. ஓகே, 16 பிளான்ட்ஸ். So 9 plants, listen, 9 plants you are getting that pure as well as then hybrid. Okay, pure alleles as well as the heterozygous alleles. And number we can have it 9 plants are round and yellow. Round and yellow. Clear? 9 plants la vandu one pure homozygous, two dominant alleles ஒரு இது. மத்த பாத்தீங்க மத்த இதுல என்ன பாத்தீங்க ஒரு dominance ஒரு recessive. Okay, dominance recessive அப்படினு. So totally we get 9 plants. We get 9 plants. Clear? So 9 plants இருக்கு. அடுத்து பார்த்தீங்கன்னா 3 plants. One dominance, one recessive. 3 plants வந்து new traits, that is an round and green color seed. அடுத்த 3 plants வந்து wrinkled and yellow color seed. புரியுதா? So out of 16 plants in F2 progeny out of 16 plants 9 plants வந்து they show the phenotypes of round and yellow. Okay, one plant show that wrinkled and green. Got it? The remaining 3 that's they are different new traits, randomly reunite பண்ணிட்டு new traits உருவாக்கி விடுது, which is not from the parents. Okay, so அப்ப நாம எடுத்த அந்த parent உடைய character எத்தனை plantsல இருக்கு? 9 plus 1. So 10 plantsல வந்து நாம எடுத்த அந்த parent உடைய trait characters அப்படியே resemble ஆயிருக்கு. ஆனா 3 plus 3, the 3 plus 3 that is 6. And the 6 plants they are independently expressed. The traits are independent. Round trait is independence of green. Round வந்து green மேல independence. Okay, the wrinkled also independence of yellow. So thereby he concluded that one trait is independent of another trait and express itself. Do you understand what I am saying? நான் சொல்றது புரியுதா? அந்த F2 ப்ராஜெக்ட்ல வந்து 16 plants கிடைச்சது. அந்த 16 plants 9 plants வந்து round and yellow, ஒரு plant வந்து wrinkled and green. இது நாம எடுத்த parent உடைய trait. Okay, homozygous trait அப்படியே இருக்கு. Okay, sorry, that phenotype traits அப்படியே இருக்கு. ஆனா இந்த 3 இருக்கு பாருங்க, இந்த மிடில் வந்து 3 round green, 3 wrinkled yellow. இந்த traits நாம எடுத்தது கிடையாது. இது மிக்ஸ், அதனுடைய இது independently express ஆயிடுது. So அந்த seed உடைய shape, அந்த trait அந்த color of the seed வந்து block பண்ணல. seed உடைய shape color of the seed வந்து block பண்ணல, but individually independently express, அதுவா express பண்ணுது. அதனால தான் நமக்கு இந்த combinations, new combinations, we are able to get the new combination, that's a new traits. Okay, apart from that our parents traits, we are able to get the new combinations, that is a new traits appeared in the F2 progeny of dihybrid. So from this point of view we will find out F2 progeny or phenotypes in F2. எத்தனை phenotypes F2 ல கிடைக்குது நமக்கு? number of phenotypes in F2 generation. Number of phenotypes in F2 generation total நமக்கு எத்தனை கிடைக்குது? 4. சரியா? number of phenotype in F2 generations. Clear? Got it? Number of genotypes. So number of genotypes only 9. So number of possible in a formula, so we will get it in 4. You understand? All the things you have to keep it in your mind. So dihybrid explain the law of independent assortment. Law of independent assortments. So what is law of independent assortments? What is law of independent assortments? So when two pair of contrasting traits are crossed, one trait is independent of another trait and expressed independently. Okay, that is law of independent assortments. Law of independent assortments. So in case of law of independent assortments, so in a pair of, when two pair of traits, two pair of contrasting, so when two pair of contrasting traits are crossed, the traits are independent of each other and expressed independently. So this is, we call it an law of independent assortments. Thank you for watching my channel.